How does the tech that runs our world really work? We break it down to its fundamental truths. This is First Principles.
There is a single word in a single sentence on a single page of a 3 billion letter book. This book is the human genome, the instruction manual for building and running a human body. And this one word has a typo, a single letter mistake. Instead of the correct word, thymine, the book reads adenine. Because of this one typo, a protein in our red blood cells gets built incorrectly. It gets built wrong. And this one tiny error causes the cells to twist into a crescent shape, a rigid sickle that gets stuck in blood vessels, causing excruciating pain, organ damage, and early death. This is sickle cell anemia. For all of human history, that typo was permanent. It was destiny, written not just in ink, but in the very fabric of a person's biology, passed down from generation to generation. And the cure, well, the cure was science fiction. How could it not be? To cure, it wouldn't mean taking a pill to manage the symptoms. It would mean finding that single, wrong letter in every single relevant cell. And what? Correcting it. How do you perform a find and replace on a living human being? But today, right now, that is exactly what we are doing. We have found a tool that can, with astonishing precision, find that single wrong letter and change it. We've given this tool a name that sounds like it belongs on the side of a futuristic appliance: CRISPR. It's a black box that promises to do the impossible. Today, we're not just going to look inside that box. We're going to break it down to its first principles. We are going to find out how humanity learned to edit the code of life. The magic of CRISPR, the magic we see, is its promise. It's the promise of an end to genetic destiny. We live in a world haunted by diseases written into our DNA. Huntington's disease, a devastating neurological decline caused by a single gene that stutters, repeating itself over and over. Muscular dystrophy, where the instructions for building healthy muscles are corrupted. Cystic fibrosis, Tay-Sachs. These are not invaders from the outside, like a virus or a bacteria. These are errors in the code within. And for decades, gene therapy was the great hope. But it was a clumsy, heartbreaking hope. The idea was simple enough. If a person has a broken gene, just give them a working copy. The problem was delivery. How do you get that new, healthy gene into the cell? And more importantly, how do you get it into the right place? The early attempts in the 1990s often involved using a virus as a delivery truck. Scientists would disarm a virus, hollow it out, and pack it full of the good gene. The virus would then do what it does best: infect the human cells and inject its cargo. But this was a shotgun blast in the dark. The virus would jam the new gene into the human genome, but at random. Sometimes it would land in a genetic desert, doing nothing. Sometimes it would land in the middle of another crucial gene, disrupting it and, in some tragic cases, even causing cancer. And in 1999, this entire field came to a halt with the death of Jesse Gelsinger. He was an 18-year-old with a metabolic disorder, and he volunteered for a gene therapy trial. The virus vector used to deliver the cure triggered a catastrophic immune response. His body attacked the delivery truck, and he died just 4 days later. The dream of gene therapy died with him for a long time. The problem was clear. We didn't need a shotgun. We needed a scalpel. We needed a tool that could not just add a new gene, but could find a specific sequence, a specific typo, and fix it right where it was without touching anything else. We needed something that just didn't exist. The world before CRISPR was a world where we were becoming brilliant genetic readers. As we talked about in our last episode, the Human Genome Project gave us the book. We could read the 3.2 billion letters. We could find the typos. We were phenomenal proofreaders, but we were powerless to correct them. We were like librarians who could find every error in every book in the library, but we had no pencil, no whiteout, no way to fix a single one. The problem, the insurmountable obstacle, was precision. The genome is not a neat 500-page book. It's a library containing 1,000 copies of a 3.2 billion letter instruction manual. Crammed into the microscopic nucleus of a cell, finding one gene is like finding one specific sentence. Finding one letter is like trying to find a single E in that entire library, blindfolded, and change it to an A. The answer wouldn't come from a billion-dollar government project. It wouldn't come from a DARPA competition or a Manhattan Project for biology. It came from yogurt and salt flats. It came from scientists who were studying something that seemed completely unrelated: how bacteria survive. The story begins in 1987, in Santa Pola, Spain. A scientist named Francisco Mojica is studying microbes, archaea, that live in intensely salty marshes. He's sequencing their DNA, and he keeps noticing something weird. He sees these short, identical DNA sequences that repeat over and over again. And, crucially, in between these perfect repeats are unique little bits of spacer DNA, which don't seem to match anything. Imagine reading a sentence like this: "The house is red spacer 1, the house is red spacer 2, the house is red spacer 3. The house is red part is the repeat. But what were those spacers? For years, it was a biological curiosity, a mystery. Mojica and a few other labs around the world cataloged these sequences. They gave them a name that only a biologist could love: clustered, regularly interspaced short palindromic repeats, or for short CRISPR. For over a decade, this was just a weird corner of microbiology, a little puzzle. But in the early 2000s, computers and DNA databases got fast enough to play a new game. Scientists could take those weird spacer sequences and run them through a massive database of all known DNA, a Google for genetics. And when they did, they got a hit, a shocking, world-changing hit. Those unique little spacers, they weren't bacterial DNA, they were viral DNA. Specifically, they were snippets of DNA from bacteriophages, the viruses that attack bacteria. Suddenly, this weird, repeating pattern made sense. It wasn't a glitch. It was a most wanted gallery. It was a collection of mugshots. The bacteria were keeping a genetic record of every virus that had ever tried to kill them. This was an immune system, an adaptive immune system. We thought only complex creatures like us had that, a system that could learn and remember new threats. But here it was in the simplest of organisms. This discovery opened the box. It revealed the first principle. Bacteria remember their enemies by stealing a piece of their DNA and storing it in their own. But a memory is useless without a way to act on it. This is where the second part of the system comes in. Alongside the CRISPR memory bank in the bacterial DNA, there's another set of genes. These are the CRISPR-associated genes, or SAS genes. These genes are the instructions for building the enforcers. They are the molecular machinery, the scissors and sentry guards of the cell. Here's how the entire beautiful natural system works. This is the real story from first principles. A bacterium is floating around. A virus, a bacteriophage, attacks. It latches onto the cell wall and injects its DNA. It's a hostile takeover, an attempt to turn the bacterium into a virus copying factory. Now, most of the time, this works. The virus wins, the bacterium dies. But sometimes, the bacterium survives, and when it does, it activates its SAS enzymes. A SARS protein finds the foreign viral DNA, snips off a small clean piece, and carries it back to the bacterium's own chromosome. It finds that CRISPR memory bank, and it pastes this new mugshot into the library as a new spacer. Never forget this guy, it says. The bacterium is now vaccinated against this virus. So that's the memory part. Now for the defense part. The bacterium doesn't just let this library sit there. It constantly reads its entire CRISPR library. It makes little copies of all those mugshots, all those spacers. But it makes these copies not as DNA, but as RNA. This is the guide RNA. Think of RNA as a sticky copy of DNA. It's a single strand, and it is chemically programmed to find and zip up to its mirror image. So, the bacterium now has all these little guide RNAs floating around, each one a perfect match for a different virus it has encountered. Each one of these guide RNAs then pairs up with a SARS enforcer protein. The most famous of these is SARS9. You can think of SARS9 as a pair of molecular scissors, or a sentry, but it's a sentry with a blindfold on. It can't find the enemy on its own. It needs a guide. The guide RNA is its seeing guide dog. The guide RNA holds the mugshot, and the SARS9 protein holds the scissors. Together, they form a search and destroy complex, constantly patrolling the inside of the cell. Now, the same virus attacks again. It injects its DNA, ready for the takeover. But this time, the cell is ready. The guide RNA SARS9 complex bumps into the new viral DNA. The guide RNA, which is that little sticky copy, frantically checks all the DNA it meets. Does this match my mugshot? Does this match? Does this match? And then, click. The guide RNA finds its perfect mirror image on the invading virus's DNA. It zips up along its entire length. This zipping action, this perfect match, acts like a key in a lock. It's a signal to the SARS9 protein it's attached to. It's the go command. And the SARS9 protein, which is an enzyme, unholsters its molecular scissors. And it does one simple thing. It cuts. It makes a clean, precise double-strand break right through the virus's DNA. Snip. And for a piece of viral DNA, a clean cut like that is a death sentence. The code is broken. The instructions are scrambled. The threat is neutralized. The bacterium is safe. This system, this beautiful, elegant immune system, has been evolving in bacteria for billions of years. A search function, the guide RNA, paired with a cut function, the SARS9 protein. For years, scientists studied this system. And then, in 2012, two scientists, Emmanuel Charpentier and Jennifer Doudna, had the big idea. They were studying the system in a lab. They realized that this mechanism was surprisingly simple, and crucially, it was programmable. They realized the SARS9 protein was a general-purpose scissor. It would cut anything. It didn't care. The only thing that told it where to cut was its little companion, the guide RNA. The guide RNA was the software that gave the hardware its target. And Charpentier and Doudna figured out that you could divorce this system from its bacterial origins. You didn't need the whole bacterium. You just needed the two components, the SARS9 scissor and the guide RNA, and this is the breakthrough. You could synthesize your own guide RNA in a lab. You could write any genetic sequence you wanted into that guide RNA. You could write a sequence that matched, say, the gene for Huntington's disease, or the gene that makes a mosquito carry malaria, or the gene that makes a plant vulnerable to drought. You could create a search string for any sequence in any organism. You could then mix it with the SARS9 scissor protein and put this two-part complex into a cell, a human cell, a plant cell, an animal cell, and the complex would do what it has always done. It would hunt, it would patrol the entire 3 billion letter genome, and it would only cut when it found the exact spot that matched its guide. This was the moment. This was the scalpel. This was the tool that scientists had been dreaming of for 50 years. This was the search and cut function for all of life. But wait, this is where we have to address the big misconception. This is where we get to the real aha moment. So we can cut the DNA. That's amazing. We can find the sickle cell typo and snip the DNA right at that spot. But how does that fix it? A cut is a break. It's damage. How do you go from search and cut to search and replace? This is the common misunderstanding. Many people imagine that the CRISPR system carries the new gene with it and just pastes it in, that the SOS9 protein cuts out the bad code and inserts the good code. That is not what happens. CRISPR on its own does not edit or replace anything. It is a search and cut tool, period. The replace part, the editing, that's the unseen giant. And the giant is the cell itself. We aren't doing the editing. We are tricking the cell into doing it for us by exploiting a 4 billion-year-old survival mechanism. Here's how. When a DNA strand in a living cell gets a double-strand break, meaning both sides of the ladder are snapped, this is not a minor event. This is a 5-alarm fire. This is a code-red existential crisis for the cell. A broken chromosome is catastrophic. If it's not fixed and fast, the cell will die or it will mutate into cancer. So, the cell has powerful, ancient, and incredibly sophisticated DNA repair crews on standby 24/7. When CRISPR makes its cut, it's like a special ops team blowing a hole in a specific wall. Immediately, the cell's repair crew alarms go off, and they rush to the site of the damage. Now, this repair crew has two different ways of fixing the break. The first way is the fast and sloppy way. It's called non-homologous end joining, or NHEJ. This is the duct tape approach. The repair crew just grabs the two broken ends and shoves them back together as fast as possible. Just seal the breach, it doesn't check for typos, it just glues the ends. This process is messy. It almost always results in a few letters of DNA being accidentally added or deleted right at the cut site. This creates a typo, and in genetics, a random typo in the middle of a gene is usually catastrophic for that gene. It scrambles the rest of the instructions. The protein gets built as gibberish or not at all. The gene is effectively broken. And for scientists, this is incredibly useful. This is our off switch. If you have a gene that is causing a problem, say the gene that allows HIV to enter a cell, you don't need to replace it. You just need to break it. You can send in CRISPR SOS9, tell it to cut that gene, and then just walk away. The cell's own sloppy repair crew will rush in, fix the break, and in the process permanently disable the gene. This is called a gene knockout, and it's the first major use of CRISPR. But this is not a fix. This is a break. This doesn't help you correct the sickle cell typo. It would just break the hemoglobin gene entirely. To correct the typo, we need the other repair mechanism. This second repair system is the unseen giant. It's the aha moment. It's called homology-directed repair, or HDR. This is the precise repair crew. This is the master carpenter, not the duct tape guy. The cell uses this system when it has time, and crucially, when it has a blueprint. When this HDR crew arrives at the broken DNA, it doesn't just jam the ends together. It says, whoa, wait, let's fix this right. We need a template. We need the blueprints for this section so we can rebuild it perfectly. Where does it find a blueprint? In a human cell, we have 2 copies of every chromosome, one from each parent. The HDR system will look at the other unbroken copy of the chromosome and use it as the perfect template to rebuild the broken section, letter for letter. And this is the process we hijack. This is the replace in search and replace. When we send the CRISPR saw's 9-inch search and cut tool into the cell, we don't send it alone. We also flood the cell with millions of tiny, separate new pieces of DNA, and these new pieces of DNA are our fake blueprints. This new DNA, which we designed and synthesized in a lab, is a short strand that is a perfect match for the area around the cut, but with one small change. In the middle, right where the sickle cell typo was, we've written the correct letter. We've written thymine. So, CRISPR makes the cut. The cell's 5-alarm fire goes off. The precise HDR repair crew arrives and starts frantically looking for a template. It sees the other chromosome, yes, but it also sees these millions of new blueprints we've flooded the cell with. And these new blueprints are a perfect match for the broken ends. The cell's repair system, in its desperate hurry to fix the break, grabs our fake blueprint. It says, "Great, here's the template," and it diligently, faithfully, rebuilds the broken section, letter by letter, copying our template. And just like that, the A is gone and the T is rebuilt in its place. The typo is corrected. The search and replace is complete. We didn't do the replacing. The Saw's 9 protein didn't paste anything. We simply made a precise cut, and then, like a con artist, we slipped a new set of instructions to the cell's own ancient and very trusting repair crew. We provided a new template. The cell did the rest. That is the first principle of CRISPR. It's not a single magic wand. It's a two-step partnership. It's a high-tech programmable scissor from a bacterium, combined with the exploitation of a 4-billion-year-old DNA repair system that belongs to the cell itself. We started with a typo in the Book of Life, a problem that seemed as permanent as the stars. We looked for a magic wand to fix it. We found a blunt, dangerous shotgun in early gene therapy. And then, by studying microbes in a salt flat, we found something else. We found an ancient war between bacteria and viruses, and in that war, we found a search and cut system that bacteria use to defend themselves. We learned to speak its language. We learned to program its search function. And then we learned that if we combined our cut with a new set of blueprints, we could trick the cell's own repair crew into doing the replace for us. We went from reading the genome to writing it. We've taken a tool that bacteria evolved to survive, and we've turned it into a tool that could change what it means to be human. This technology is no longer science fiction. It is being used right now to cure people of sickle cell disease, to edit crops to survive climate change, to potentially wipe out malaria. It is also a tool of unimaginable power with ethical questions that will define our century. We have found the search and replace function for the code of life. The question is no longer can we edit the book. The question is, what story should we write?
You've been listening to First Principles. Until next time, stay curious.